because I already recorded this once, but then realized that you could only see half the screen, which is why everything's already written out. So in this video, first question is, I'll sort of emphasize it to make it stand out a little bit here. What would happen if RNA polymerase transcribed both sides of the DNA? If it transcribed both the template and the coding strand at the same time? It only does one. Remember, it only transcribes the template strand. What would happen if it were to do both? Then, what is the region that is 3' of the transcribed region? So this region has its own name, and then within it is another special area that it contains. So what are those two names in that 3' of the transcribed region? And then what do upstream and downstream mean? How do you determine what is upstream and what is downstream? And where does RNA polymerase bind versus where does it actually start transcribing? And finally, what are the stages of transcription?
Okay, so now that we've established kind of the major actors of transcription, we're going to step through a generalized description of how transcription proceeds, focusing primarily on eukaryotes. All right, so I showed you this image because it's important to remember that what we're talking about here is a double-stranded piece of DNA. So this white area represents the promoter, the green area represents the consensus sequence, and then the yellow represents the transcribed region. For the next couple of steps, I'm going to show you a sort of simplified version of this, but keep in mind that we are still talking about a piece of double-stranded DNA. So this is the sort of simplified version. Oh, and I need to fix that. So it's the same sort of information, just a slightly different image, still indicating to you that we're talking about transcription. All right, so here we have the promoter region. Here is our gene sequence to be transcribed, and so we're going to go through the steps, the generalized steps of what happens when a gene is going to be transcribed. So this little section right here is called a TATA box. And for many genes in the eukaryotic genome, this TATA box is the consensus sequence that RNA polymerase needs to tell it this is where you start. And the TATA box, for some reason it's called a box, I'm not exactly sure why, but it's a sequence of T's and A's. So it's T, A, T, and then a bunch of A's, A. So on the other side, of course, it's a bunch of T's. And if you remember, between thymine and adenine, you only have 2 hydrogen bonds. And so this is a relatively weak area. And so when RNA polymerase comes and it starts getting ready to transcribe, it's easy for it to separate the 2 strands of DNA here at this TATA box. So what happens when this gene is going to be transcribed is that this protein called a TATA binding protein, and it comes and it binds to the TATA box. And then a transcription factor comes and binds to that TATA binding protein, and you have to have both of those. So they come and they bind to the consensus sequence embedded within the promoter region. And then what that does is it recruits RNA polymerase to come and bind the DNA. So you've got to have both the binding protein and the transcription factor bound at the correct location to recruit RNA polymerase. Okay, so then what RNA polymerase does is it unzips the DNA, which is so cool. It unzips the DNA, and then it starts adding the nucleotides. So remember, these are ribonucleotides, not deoxyribonucleotides. And RNA polymerase is so cool because remember when we did DNA, and we were talking about how we replicate DNA, we had to have helicase, and then we had to have, a primer, primase come in, and then we had DNA polymerase come in. But RNA polymerase does all of that. So in this sort of first walkthrough of the steps of transcription, we're going to talk about RNA polymerase in just kind of a simplified, like here's RNA polymerase as if it's just this one thing. But keep in mind that it's actually this crazy complex. So this is sort of an image indicating what it actually looks like molecularly. And so you've got all of this different structures. So you've got alpha helices and beta helices all together, and then this green stuff is the DNA coming through it. And then this is an artist's depiction of all of the different things that come together, all the different proteins and cofactors and activators and repressors that come together to actually make this transcription process work. So for right now, as we're going through the generalized description, you're just going to kind of see this lump that represents RNA polymerase. But keep in mind that it's vastly more complex than that, and we're going to be talking about that in more detail when we talk about transcriptional regulation. Okay, so initiation, what this is initiation. So initiation is when RNA polymerase begins unzipping the DNA, and it starts creating, and it can do this from scratch, so it doesn't need primase, it doesn't need a helicase. It starts creating the mRNA just from scratch using ribonucleotides. So RNA polymerase, here's that in words, both unzips the DNA helix and interacts with the 3' to 5' strand. So DNA, we know, of course, is double-stranded. RNA polymerase, the strand that it interacts with is this one, the 3' to 5' strand. So an important point, and I'm going to show you a visual of this in a minute, is that RNA polymerase is only creating an mRNA, a complementary mRNA, to one side of the template. So it follows the 3' to 5' strand because, like RNA polymerase, it can only add to the 3' end of its growing mRNA. So it still needs that, that end on the pentose sugar so that it can add the phosphate to the growing 3' end. All right, so let's look at this in a little bit more detail. So let's add bases to our strand. So if this top strand, this is our imaginary sequence, and this bottom strand, this is our imaginary sequence. So as RNA polymerase is adding bases, what it's creating is a strand that's almost identical to the other strand of DNA. Because remember, they're complementary, right? They're anti-parallel and complementary. So if we look at what mRNA is producing, it's producing a strand that almost identically matches the complementary strand of DNA, right? The difference is that there's a uracil instead of a thymine. So if RNA polymerase was doing this on both strands, so like how DNA replication occurs on both strands at the same time on the leading and lagging strand, if RNA polymerase were doing this, so let's say this is what's actually happening, is RNA polymerase is synthesizing a strand, adding to the 3' end, moving on the template strand from 3' to 5'. So here is the other strand. If RNA polymerase were also doing the same thing on this strand, moving, adding to the 3' direction, then what it would be creating, it would be creating two strands that are perfectly complementary to each other. And what would happen is that those two strands are going to bind to each other. So that adenine is going to find that thymine is going to find that adenine, and on down the way. Oh, I did that wrong, guys. I apologize. Hang on, I need to fix that. Okay, fixed it. So adenine would be binding to the uracil would be binding to the adenine, guanine would be binding to the cytosine. And the thing is that these sequences, these mRNA sequences would be perfectly complementary to each other. So what you would be doing if you were creating an mRNA on both sides of the DNA is you would be creating perfectly complementary sequences. They would bind to each other, and then they wouldn't be able to leave the nucleus. They wouldn't be able to be processed. They wouldn't be able to leave the nucleus, and they wouldn't be able to be translated into proteins. So the RNA polymerase only transcribes one side of the DNA at a time. For that reason, and also because it can only transcribe the DNA, remember, where it finds a promoter. So we've got to have a promoter, and that has a very specific sequence. And so it's looking for a specific sequence of the promoter, of like the Tata box, for example, and the flanking sequences. And so if it finds the promoter on this side, then it transcribes this side. Had it found it over here on this side, then it would transcribe from this end to this end. So different genes are transcribed on different sides of the DNA. And this is another active area of research where there are some cases where we don't know exactly which side of our DNA is being transcribed and then translated. Okay, so we have our promoter region, our consensus sequence. In this case, we're discussing the Tata box. We have our Tata binding protein and transcription factor. They recruit the RNA polymerase. So the RNA polymerase, then it finds the Tata box, and then it sort of ratchets down the rest of the promoter region until it finds what's called the +1. So the +1 region is where RNA polymerase actually begins synthesizing the mRNA. Everything behind the +1 region, +1 base actually, is called upstream. Everything after it is called downstream. So this is more terminology. Okay, so we initiate. This is when the polymerase unzips the DNA and begins creating the mRNA transcript. The next step is just elongation. So this part is just super simple. It's just making that mRNA transcript longer. The last part is also pretty simple conceptually. It's termination. So the RNA polymerase reaches a region of the DNA that's called the terminator region. And when it reaches that terminator region, it terminates transcription and releases that mRNA. So there are several different ways that termination can occur, and it depends on which RNA polymerase is doing the transcription. But basically, most of the ways is that the terminator region slows RNA polymerase, so makes it move slower, and so it's more likely to dissociate. And in some cases, the terminator region recruits another protein, and that protein inhibits RNA polymerase from continuing forward. So basically, the bottom line is that there's a specific sequence of DNA down here, and it causes RNA polymerase to dissociate. And when RNA polymerase dissociates from the DNA, it releases the mRNA transcript. So we're focusing on eukaryotic transcription. Just want to show you here the transcription process in prokaryotes. Super similar. That's there for you. And then again, showing you, this is an actual real image of transcription in prokaryotes. So remember, prokaryotes don't have that nucleus. And so what's happening is you've got the transcript. This is the DNA right here, this little line. And the transcript is being made. It's coming off the DNA. It hasn't even been released from the DNA yet. And you've already got a bunch of ribosomes all clustered on to that transcript. They're already translating it into protein. And when we get into translation, you might see why that's kind of cool and weird, a cool thing about prokaryotes. All right, so that's it for this video. We will talk about transcriptional regulation next.